Thank you so much for this kind invitation. I'm deeply grateful and honored by the invitation by the Endocrine Society of India. I've heard the first presentation just before I came into the connection, and I was really very much hurted by the quality of the data and the nice presentation. Well, so we're gonna start with this, I would say it's still hot topic, which is the personalized treatment of acromegaly. This is my disclosures. What is personalized medicine? I will just like to remind an important, interesting paper which came to the press in the New England Journal of Medicine about 5 years ago. And regarding precision medicine, the epitopes that were used were personalized, but also problematic as well as promising. Just reviewing what does it means, personalized or a similar concept, which is precision medicine, which has been very recent in the last years, either from the scientific, but also from the, let's say, media and political aspects, because you might remember that President Obama made a large contribution of money for personalized and precision medicine in brain research. What we really try to go with this concept is that we are assuming that most of the diseases are not etiologically unique, but are mostly syndromic. And what we are trying to do is to stratify or to divide or classify much betterly our patients in order to have a much better and targeted therapies. This means that we are entering in this concept of deep phenotyping.
Everybody please mute. Everybody please mute your audios. I'm sorry, Dr. Manuel.
Okay. So we are trying to stratify our patients in order to get much more accurate treatments which will be more effective. Regarding acromegaly, do we really personalize our treatments or not? And I think that we all share the idea or the concept, we do agree that the pharmacological, at least pharmacological treatment of acromegaly is currently still based on the concept of assay error strategy, in which you try a drug, and if it doesn't work, you try another drug, and if not another, or you combine, and so on until you get what it's working for the patient. But this could last years, even years, in certain patients. And we know that most clinical guidelines recommend somatostatin receptor ligands as first treatment of acromegaly when we use drug for the patients. And we know also that about 50% of these patients do not respond adequately to these drugs. If we just go for a old revision by Pamela Freda in the JCM, you can see here in brackets that most of the patients do not meet the criteria of, and this is an old number, 2.5 nanograms per liter of GH, or normalizing IGF together with reduction of tumor burden. If we would even combine these three items, the results of the patients which are controlled are even lower. We also did this exercise more than 10 years ago, and we ascertained that our patients in 2 large hospitals in Barcelona were not controlled regarding IGF-1 normalizations in more than 40% of cases after 1 year of treatment, either as adjunctive or primary therapy. So the question is, if the same disease is active in the same patients, if this is not the case, which is what we now know, it means that we could not use the same treatment for all the patients. And we proposed in 2015 that it was time to move on and to go for being much more according to what is the current era of personalized and predictive medicine. This was followed by different articles, and I would like also to recall this one by Sylvia Asa and Sherin Esad, in which they clearly describe that acromegaly is presenting as a spectrum of morphological pituitary tumors, different between them, showing different clinical, biochemical, and physiological features, which also require different therapeutic outcomes. And this is due to the background of the genetic and epigenetic nature, which is not the same. And thus it was, they proposed that it was time to go probably for the era of increasing the diagnostic, meaning make a diagnosis in which it was included information which would predict or would help the clinicians at the time of decision of treatment. Also, another prominent pituitologist, which is Monica Gadelha, Brazil, indicated in a paper of the European Journal of Endocrinology in 2018 that this would much quicker disease control, and obviously, beside the personal health cost, also a reduction of the cost for the patients. However, all the clinical guidelines we are currently are still giving you the quiz of what to decide if to go for one or another modality of the treatments. And you know, as well as me, that we will be having increasing number of drugs for acromegaly treatment, which will pose even a more challenging situation at the time of the decision of what the treatment would be best for a given, for a special one person, single patient. This is just to exemplify that the clinical guidelines, this is an old one, even in the big meetings in where consensus are tried to get the light. If you see what are the algorithms, the word which is more frequently included in this algorithm is not the word control, but the word uncontrolled. Our patients has to suffer uncontrolled, 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 and more uncontrolled situation. This takes time. So the key is to select the patients as much as possible, as accurate as possible, in order to get if they will be responsive to this treatment or they will require an addition of drugs, so should be best put on a GH receptor antagonist, namely prednisone, after diagnosis. And this is what we should currently answer is these two questions. First, if we are able to predict therapeutic response before the surgical treatment, in the case we will give the patients medical treatment before surgery. And in the case that the surgical treatment has failed, if we have enough information to decide what will be the best pharmacological adjunctive treatment for these specific patients. Let's go for the first question. And my personal answer in this case is yes, or probably yes. And I will explain you what are the reasons for giving this a positive answer. The first, we all perform in our patients a diagnosis is a nuclear magnetic resonance imaging, in which we can currently define at least 3 different patterns when we are assessing the T2-weighted signal. And it's relatively easy to categorize by any skilled neuroradiologist that the images, when compared to the cortical adjacent parenchyma, could be either hypointense, hyperintense, or about the same quality of intensity signal. And we described more than 10 years ago that this particular feature, meaning the hypointense signal, was identifying a large number of patients that show a good response to first generation somatostatin receptor ligands. And as a mirror situation in those depicting hyperintense signal, they were more prone to have a non-responsive situation. And this was posteriorly confirmed by different studies in which not just this clinical radiologic concordance was shown, but also that was enriched with pathological data indicating that these patients depicting hypointense signal have specific pathological features that I will explain later on. But also, and lately, we have seen that even those patients in which the hyperintense signal was found in their MRI, they were also more prone to be responsive to pasirreotide, one of the latest multi-ligand compounds for somatostatin receptor interaction in acromegaly patients. So we have now enough information that could help at the very beginning of the diagnosis stage, meaning the information that we have when we perform an MRI. Not just that, but we can also functionally try to characterize these patients by the use of the all described acute octreotide test. We also performed a study in 2008, and we were able to see that most of the patients after diagnosis, when they were given 100 micrograms of regular somatostatin subcutaneously, in most of the cases, the nadir of GH was observed at about 2 to 3 hours after injections. And when we correlate this, what was happening by using maximal doses of first generation somatostatin receptor ligand, we show that the correlation was quite consistent with the Spearman R of 0.76. By using this approach, we were able to define different cutoff in which we were identifying when the nadir was below 3.6, those patients that were completely responders, and when the nadir was up to 2.7, 7.2, those patients that were really resistant. And in between, we defined what we call the partially responsive patients. This has been very useful, and we have now re-evaluated by the application of the new last generation ultrasensitive GH immunoassays, and now this 3.5 cutoff has gone down to 2.7 nanograms per ml. This has been lately revisited by a group of China researchers, in which they also performed a much deep study in which they also included MRI and the different signal in T2, and they were also concordant with more or less what we got, which is that the nadir GH of about 3 is very informative for identifying responsive individuals. But not just that, but with a more sophisticated approach, they showed that the percentage fall of the GH nadir was the best predictor for identifying those patients that were prone to respond completely when using somatostatin receptor ligands of third generation. This is why we proposed already in 2015 that the addition of these two informations, which are very easily useful in the moment of diagnosis, would allow us the combination of a patient who would be very prone to good response with somatostatin receptor ligands as monotherapy, when we got a hypo-intensity to signal together with a lower than 3 nanograms per milliliter GH after an acute octreotide test. And those that were having hyperintense plus higher than 7 nanograms per ml, they were clearly much more feasibly and effectively treated by the monotherapy, but with pasirreotide or perpizoman, and in between the combined treatment was a reasonable approach. Okay, now going for the second question is, we have a patient in which we are not being able to cure by surgical treatment. Could we predict him with the addition of the information we got from the tumor after excision of this tumor? And my answer in this case is maybe. How could we do so? We could do so, obviously, studying what is expressed at the molecular level with the tumor, which we have at least removed partially. And you might see that in the last 10 years, we have a plethora of information in which different molecules have been involved as identifiers of response of medical treatment in those patients after surgical failure. and beta-arrestin families, together as E-cadherin, of course, somatostatin receptor subtypes, have been of a lot of helpfulness in order to define what will be the molecular profiling that could help to identify those patients in which these treatments could be effective or not effective. It is difficult, and we have assisted at many, more than a dozen different biomarkers, because the response to somatostatin analogs is driven by different molecular ways. One is the somatostatin receptor family itself, but all the mechanisms that are involved in the post-receptoral pathways that are activated or deactivated after somatostatin is linked to its family receptors. And you can see here that depending on the combination of the activation of these different pathways, we can get all the responses we are expecting when we're giving these drugs, either regarding hormonal control, but also cell proliferation, meaning the action upon the reduction of the tumor size when giving these treatments. And we have seen that in most of these cases, when we focus on single biomarkers, we might show interesting results, which can explain us how the disease is behaving. And regarding somatostatin receptors, it has been consistently described that the combination of the abundance of STST-TR2 and 5 and its ratio particularly seems to be a good predictor. A good predictor that could explain how most of the patients behave. But as you can see, the cutoff levels are quite interacting, and there is no separation. There is some overlapping of this result, which make difficult to use this single information for a single patient. This is why, together with the technology used for measuring these receptors, has made or has led us to the conclusion that not always all these results are concordant and the correlations are not always consistent. Moreover, we have to have experienced immunohistochemists, pathologists, which should score and to localize where these receptors are expressed, either in the cytoplasmic or at the membrane level, which makes the difference. And so we need to standardize how this method is applied and to refer in order to get information which will be comparable between centers. Moreover, it has also been described for these tumors that the granular pattern assessed by the cytokeratin 5.4 is very informative, and we can see different patterns. The more extremes are those of sparely granulated or densely granulated pattern, with in between them some different patterns. And it has been described that these patterns are very much predictive with an odds ratio of about 60 regarding their responsiveness to somatostatin receptor ligands. This is very important together with tumor stage under Knopf's category, and this has also been correlated with the expression of the somatostatin receptors. But again, as you can see here, the level of distribution of the values regarding the granulating pattern is very heterogeneous between patients, and only somatostatin receptor 2 is really linked to this specific densely granulated pattern. We try to overcome this question by making a broad molecular profiling analysis by using more than 100 patients of Spanish cohort, in which we use those that were resistant or complete responder or those in between. And we measured all the different markers that have been individually described as apparently defining which patient will be responder or not. And what we got here is first that when applying all this information, we confirm the difficulty of treating these patients because all the tumors are showing in between patients a very heterogeneous conditions. You can see here how are depicted different profiles according to the tumors and according to the different patterns. However, some clustering seem more or less possible, which also is more or less coupled to the different phenotypes, meaning that it's not the same to be old or young. It's not the same to be a giant or not a giant. So this is the difficulty we have. And when we are trying to apply what markers could be really predicting markers, those that really resisted were somatostatin receptor 2, E-cadherin, which was able, E-cadherin, to differentiate between complete and partial responders and also resistant responders from a statistical point of view. But again, you can see that dispersion of the data found, it's clearly very important, and the overlapping in between subjects and in groups it's really very high. And the receiving operating curves are not upper than 0.75 in the best case, which is the one of E-cadherin. Moreover, Ki-67 was also informative of those that were non-responders in comparison to complete responders. But even in this case, we have some patients that scale and show high level of Ki-67, even if the response to these drugs is very good. When we tried to pull those markers that were more informative, you can see that the combination of E-cadherin and somatostatin receptor 2 addition was not better than what E-cadherin showed per se, remaining at a sensitivity level of 70% together with specificity and a positive and predictive value, which is around 70%. This means that all the data, all the markers we try to explore are probably not the complete family of markers that could explain a single personal response. Moreover, when we perform a correlation Spearman matrix, you can see that all the markers used to be related in between them, each other, and some of them show important correlation like it was expected, somatostatin receptor 2 together with E-cadherin. So for overcoming this and not just having information which is able to explain the disease, but not the disease in a patient, we have to go for a different methodology. And the one should be applied currently is those that is based in artificial intelligence, and specifically data mining. In data mining, what we are doing is to have an approach combining artificial intelligence together with systems biology, with the interaction of machine learning processes with classical statistics. And we are able to combine different biomarkers and to see how this will apply for a single patient, trying to get as much accuracy as possible, which is the clue for having the best prediction for a single patient. In this case, what we did here is to use our cohort of more than 100 patients, first to apply classical statistics and thereafter to include the different markers that were resisting the classical statistic analysis. And we were able, through this procedure, to get much better information for a single patient when we were fragmenting the population regarding different clinical situations. If the treatment was performed before surgery, if the patient was having extracellular growth or sinus invasion, regarding also gender, and also depending on certain mutations which are also linked to good or bad response, age, and the different molecules that has been so far described. And through this approach, we were able to build cutoff, which were much more able to separate most of the patients that were responsive by using a combination of different of the markers that has been described. And it was even difficult to me to understand how a cutoff could be drawn this way, or we are having double or triple cutoff values when we are combining these different biomarkers. But this is what our statisticians have been able to draw after this analysis, which is now under a validation process with a different cohort. But with this way, we are able to define with the algorithm, I am finishing, what happened in the case when a treatment, surgical treatment is failing, in which the accumulation and application of artificial intelligence methods allows us the possibility with accuracies that goes up to 95% or 80%, which is higher of what we have until now, to predict what's going to happen. For example, in the case we're having a patient with an extracellular growth. In the case this patient is not having an extracellular growth tumor, we are able, with a combination of RAF kinase and somatostatin receptor 5, to define which one of the patient which will show a complete or a partial response. And in the case that we have an extracellular growth, by using ghrelin gene and somatostatin receptor 5 in ghrelin variant gene, we will be also able, with 80% of accuracy, to have what will be the pathway which will help us to, with a more precise way, to distinguish between those that will respond or not to a given treatment. This is also the same when we apply this according to the gender. But you can see here by using age and ikaderin in the case of males. And at this accuracy level, we are also able, with the addition of dopamine receptor 2 and ikaderin, what are the patients which will show, with the cutoff defined previously as I show you, which will be the patient responsive. And also in the case of female, by the use of RAF kinase inhibitor protein, ghrelin gene, and somatostatin receptor 2. And now we are just performing a prospective trial in the society, the Spanish Society of Endocrinology, with a much simple approach by using of ikaderin, somatostatin receptor 5, and somatostatin receptor 2 together with dopamine receptor 2. We're trying to see if this will allow us to get a much faster effectiveness of the patients compared with standard care. So for concluding, my messages are that precision or personalized medicine, this is interchangeable, is necessary for our single patients. Nowadays, we are able to much better subclassify somatotropinomas by using functional tests as the acute octreotide test, bioimaging, meaning magnetic resonance imaging, and histopathological combined with molecular information of the tissue sample for defining which patient will be more responsive to our current treatments, pharmacological treatments. So we are approaching the precision medicine, but still we have some way to do. This is my group in which I would like to clearly identify those that are working in the bench, Joan Gil and Mireia Jordà, and I appreciate your time, and I kindly express my gratitude for the invitation. This is a picture of my campus, and Barcelona is just here. Thank you so much.